Hi everyone. In this video, we will discuss nucleic acids. When we talk about nucleic acids, they are important in case of cell biology, in case of molecular biology. Whenever any kind of subject related to biology, you need to understand what are nucleic acids. This is just an introductory video where I will discuss the introduction about DNA, RNA, transcription, translation. I will not go into the details of these processes. I will cover these topics individually in my future videos. As you can see on this particular slide, I have tried to discuss about the bases, about the monomeric forms of DNA, what is DNA molecule, and how you can use complementary matching to detect the DNA molecule. In this video, I will just touch base on this particular topic, and if you like these kind of videos, then please subscribe to the channel, and if you like the video, hit the like button. I need your support, so please do subscribe to the channel. All right, let's start the video lecture. In this video, we will discuss the topic of nucleic acids. We're going to discuss DNA in more detail. We will also touch base on ribosomes, proteins, some of the functions of the processes that are involved in the protein formation. So first, let's discuss what is the genetic information. Genetic information is the coded information that is stored in the DNA molecule. And what is the DNA? It's the deoxyribonucleic acid that stores that information. And its structure is basically, it is made up of two strands, and they are joined together. And this joining is very strong. So here is the structure of DNA molecule. And my second point is basically DNA is composed of two helical strands, and they are wrapped around a common axis. And that's how they construct the double helix structure. And this is your double helix structure in yellow color, right? And what is the monomer of DNA molecule? You have nucleotides. Bases. These bases are basically why they are called bases, because they resemble cyclic organic bases. And you have four important bases: A, T, C, G, adenine, thiamine, cytosine, guanine. You can clearly see here adenine forms double bond with thiamine, and then cytosine, that forms a triple bond with guanine. This is complementary matching, and it's very strong. For some reason, if they get separated, they have the ability to join again. And this property, because of this property, there are so many tests that are standardized that can detect the second strand. If you have a known single strand, you can use that to detect the second one. Let's say if you have one strand that is attached to a substrate and you have second strand that you want to detect, if they're complementary, they're going to bind together. And if you label that strand with a marker, a fluorescent dye or something like that, you can detect if that particular molecule is bound to your target DNA sequence. This was just an example. Next, what is a gene? Because when we discuss DNA, we need to discuss gene. Gene basically is the sequence of DNA which carries the information. It can be very long, it can be very short. In case of bacteria, you have smaller number of genes. In case of humans, you have larger number of genes, around 20K to 25K genes. And when you have so many genes, you need to make proteins from those genes. And the process that makes the protein is transcription and translation. What is transcription? Transcription is basically from DNA, you get the formation, or you convert the DNA, the information stored in DNA, to RNA molecule. And what is RNA? RNA is ribonucleic acid. And RNA is constructed by RNA polymerase, and it's a large enzyme. Next process is the translation. Translation is basically conversion of RNA to proteins, and in that case you have ribosomes, and the role of ribosomes is to read that genetic code, universal genetic code, which is present in the RNA, but it was originally present in the DNA. So basically, this is description of the central dogma, where you have DNA sequence which gets converted into RNA, and then that gets converted into protein sequences, and then protein gets folded. That determines its function. It's really important to understand these basic processes. We'll discuss more in detail in our future videos. So if you like the video, then please do hit the like button and support the channel. Thank you. I hope that this video was helpful for you to understand the nucleic acids. We have also touched base on some of the important processes like transcription, translation. I will cover these topics in my future videos, and I hope that this video was helpful for you to start up your understanding of these processes. All right. With that note, I will request you to subscribe to the channel and show your kind support. Thank you. I'll see you in my next video. Take care.